Good afternoon, everyone. My name is Joaquin AlvarezNorambuena. I'm a first-year PhD student in the BMed program, and today I will introduce you to experimental in vitro evaluation of pairs-modified live vaccine and wild-type virus sequence detection in co-infections. So first, let's talk about pairs. What is pairs, and what is this so important to the swine industry? So first, this is a single-strand RNA-positive envelope virus. It's an arteriviridine, and its analysis came basically from the OR5 sequence, the RFLP, and the whole genome sequence. As you can see over here, the OR5 is a structural gene, and here is the actual virus. So why is this so important? Well, first because it's high cost in the swine industry. It actually counts for $0.6 billion a year, which is a lot of money. And we know that there are vaccines that we can use to mitigate the impact and reduce the shedding of the wild type infection. So that's why it's used as a control strategy. But this can be a double-edged sword, and we can take a look at why is this so important during this presentation. Also, we know that maybe sequencing might favor one strains over another, and also that the variation among years is high, and that's why they got like rapid turnovers among years. Therefore, our hypothesis is that during a PIRS outbreak, when we vaccinate, we can actually mask wild type infections. So we might not have an actual picture of what's going on in the field. And the objective of this was to assess the likelihood of detection of wild type and vaccine sequence in vitro samples when strains were co-circulating at different concentrations. For this purpose, we used two viruses. First, the wild type virus, which was an RFLP 174, and a vaccine virus, which is an MLB RFLP 252. This was grown in Mark 145 cells and supplemented with 2% FPS in DME and media. So as you can see over here, we use four different dilutions that were 10 to the 7, 10 to the 6, 10 to the 5, and 10 to the 4 RNA copies per ml. And these were mixed in different groups, considering vaccine and wild type. So these dilutions were vaccine 10 to the 7 and wild type 10 to the 7, 10 to the 4. After that, vaccine 10 to the 6 and wild type 10 to the 7, 10 to the 4. Vaccine 10 to the 5 and wild type 10 to the 7, 10 to the 4. And finally, a vaccine 10 to the 4 and wild type 10 to the 7, 10 to the 4. So what were the results of this is that we observed that that almost 70% actually of the groups, 11 from the 16 groups, were 100% vaccine. One of these group was actually 100% wild type. But as we can see over here, only 3% both vaccine and wild type in detection on the sequence. And one of the groups was actually insufficient of the genetic material for the sequence, so we couldn't assess that group. So here we got that the wild type virus was only sequenced at the 10 to the 7th and 10 to the 6th dilution, while vaccine virus was in 10 to the 5th. Also, we observed that in 10 to the 4 vaccine and 10 to the 5 wild type concentration, only one of the samples that we were evaluating got actually sufficient genetic material, and it was a wild type. Also, in 10 to the 4, they were not insufficient material, so we couldn't account this group for the study. So what are the implications in the field for this? Is that here we got the negative peaks, the negative hertz. Over here we got provisional negative, positive and stable, positive stable with vaccines, which is this orange part over here. The pink is with positive stable with live virus inoculation, and these are the positive unstable, the red ones. But when we account for the positive stable with vaccination, we may mask a wild type infection. So we need to get further steps on what is actually going on in the field and how to account for this important part in the swine industry, because we might be masking an actual wild type, saying that it's only a vaccine. That is so important. Here you can see all the reference, and thank you very much for listening to this presentation.